And Dr. Bae, he is at the Catholic University of Korea, and he has really been a pioneer in doing many, many studies that are now being published and that have been published. And so I love to learn from him, learn from his lectures, learn from his papers. And so he's one of these centers that really puts a lot of effort into answering some of these fundamental questions in vitiligo. So today we're going to hear from Dr. Bae on the development and validation of the fingertip unit for assessing facial vitiligo area scoring index, which is important, especially as we move to studies in which we're looking at the F-VASI or facial VASI, because this area we're looking for small changes. And so going from the hand unit to the thumb unit to the fingertip unit can be a great progress in our assessment methods. So I'll turn it over to Dr. Bae.
Yep. Thank you for introducing me. Hi, everyone. Did you hear me?
Yes, we hear you. Yes,
okay,
we can.
Thank you. Yeah. My name is Jung from the Catholic University of Korea. It is a great pleasure to attend this wonderful conference. First, I would like to thank all committee and managing members, including Dr. Hamjabi and Patrick, who prepared this conference. My topic today is development and validation of a fingertip unit for assessing facial VASI. There are several tools for evaluating vitiligo. They are being used to assess vitiligo extent, vitiligo disease activity, and target lesion assessment. Among them, the two most widely used and validated tools for evaluating vitiligo extent are Vitiligo Area Scoring Index, VASI, and Vitiligo Extent Score, VES. VASI is calculated as the sum of the product of a hand unit of each body part and the extent of residual depigmentation. VES is an intuitive and evaluation tool based on clinical pictures that mimic the natural distribution of vitiligo. It includes 19 body areas reflecting 6 degrees of involvement. Today, let's focus on facial vitiligo. In real life, many vitiligo patients are primarily concerned with their facial regions more than with other regions. In addition, some treatments have shown dramatic responses, especially in facial regions, and which has a very remarkable impact on the patient's life. So, facial vitiligo should be given more attention and needs to be evaluated separately from the body involvement. And then, how can he measure the vitiligo extent of this patient? Should he evaluate it with VASI? It seems a bit difficult to assess facial regions with the hand unit. The hand unit is too large to evaluate tiny regions on the face. What about VES? It is easy to assess the exact involvement, but it seems harder to distinguish the small regions on the face. Moreover, if this patient is getting better with proper treatment, it would be difficult to quantify the treatment response using VES as well. What about using our fingertips for the evaluation? The fingertip unit is intuitive and easily understandable for both clinicians and patients. Here the study began. This is an overview of our study. The study was conducted in 4 stages: determination of the fingertip unit, development of facial VASI using the fingertip unit, pilot study of the facial VASI with patient pictures, and validation study with live patient. First, determination of the fingertip unit. 98 volunteers were enrolled in Suwon, Korea and Detroit, Michigan. The palmar aspects of their hands were photographed. The surface area of the hand, the middle 3 fingers were measured respectively. The ratio of the hand unit to the fingertip unit was calculated for each patient. We found one hand corresponds to about 32 fingertip units. The ratio of the hand unit to the fingertip unit did not significantly differ among different races. Furthermore, the ratio did not significantly differ among different age groups and sex as well. As a result, we defined one fingertip unit by converting one hand unit to 32 fingertip units. Second, the development of facial VASI using the fingertip unit. Facial VASI was defined as the sum of all fingertip units needed to measure all vitiligo regions on the face. We further converted a fingertip, a finger, or a hand into the fingertip unit. We can use them to assess the area of each patch. If repigmentation occurs, facial VASI can be calculated by the sum of the product of the fingertip unit of each patch and the depigmentation of level. The depigmentation level can take a value from 0 to 1, either by a rough estimate or by selecting the most similar one from the reference images. Let's go back to the first picture. How can we measure the vitiligo extent of this patient using the fingertip unit approach? In my opinion, it appears to be around 2 fingertip units. What do you think of the involvement after 2 months treatment? In my opinion, it appears to be 0.5 fingertip unit. Third, we conducted a pilot study of the facial VASI using the fingertip unit with patient pictures. 41 dermatologists were recruited in Suwon, Seoul, and Detroit. 2 sessions of facial VASI evaluation using the fingertip unit were conducted based on the pictures of 17 patients with facial vitiligo with a 2-week interval. All assessments were conducted online using Google Forms. In the pilot study, the facial VASI using the fingertip unit showed high accuracy, intra-rater and inter-rater reliabilities. The last round was the live patient evaluation. 2 sessions of live patient evaluation were conducted with a 2-week interval. 6 raters evaluated facial VASI using the fingertip unit on 11 patients of facial vitiligo. Accuracy and inter-rater and intra-rater reliabilities were assessed by calculating concordance correlation coefficient and intra-class correlations respectively. In the live validation study, facial VASI showed remarkably high accuracy and the smallest detectable change was 2.2 fingertip unit. Facial VASI also yielded remarkably high intra-rater and inter-rater reliabilities. This is the summary of the result: accuracy, intra-rater reliability, and Bled-Altman plot. In conclusion, the fingertip unit can measure a small surface areas more precisely and easily in patients with facial vitiligo. Facial VASI using the fingertip unit is a practical and precise measurement tool for assessing the extent of facial involvement in patients with vitiligo. It can be used to quickly assess the surface area in clinical practice and also to enhance the quality of clinical research. This is the last slide, an example. Let's assess the vitiligo extent of his face using the fingertip unit. 6 plus 6 fingertip unit in the forehead, 2 fingertip unit on the inside of both eyebrows, 1 and 1 fingertip unit. All in one, it's roughly 17 fingertip unit. In the digital image analysis, it was 16.6 fingertip unit. Which one do you prefer to describe the vitiligo extent of this patient? 0.51% BSA or 17 fingertip unit? I bet on 17 fingertip unit. Whole numbers are more natural than decimal point less than 1 point. Thank you for your attention.
Thank you, Dr. Bay, for that excellent presentation about this advance in measuring vitiligo. Um, I think it's exciting to see the fingertip units, especially as we're measuring small areas. We do have time for one question, and so since I don't see any questions in the box, I would like to ask you a question. So when you are doing this huge study on photographs, and I believe that when there is a central rater looking at photographs and trying to do a VASI score, and you compare that to raters who are doing the score live, there's a 20 to 25% error. And the reason is because the photographs are usually not as good as yours. You have excellent photographs. You can see the images very well. But there is a difference between looking at the patient live versus looking at photographs. You have provided the hand of the patient holding her hand next to the face, and then the evaluator, the investigator, can see the fingertip, and they're supposed to use that and carefully with their eye, supposed to do that on the picture. That is very difficult to do on a picture. So you did it with 11 patients with 6 raters, and you showed an extremely high correlation, but you did not show any kappa values. So it makes me wonder, should something like this really should be validated with photographs? Isn't photography and rating just on photography and something we are going to do on live patients? Isn't it not the right method? Shouldn't we be doing all of these validation studies on live patients?
Yeah. Exactly. In my experience, it is more easy to assess you with the live patient more than with the patient pictures, because patient pictures did not reflect whole the aspect. But I think live patient assessment is more easy and more precise.
Yes.
And in the clinical practice, actually, I use my fingertip unit after comparing my fingertip unit and the patient fingertip unit, the size, and then usually I use my fingertip unit here and here, here and here. So I think it is very not difficult to assess the whole face.
Yeah, I agree. I think the fingertip unit is a good advance. But the PASI, the EASY for atopic dermatitis, the PASI for psoriasis, the MASI for melasma, the PAPI for post-inflammatory hyperpigmentation, all of those were validated using live patients, not photographs. So I'm glad that you did the live patient rating. Thank you again for your presentation.